Okay, today we're going to be talking about cells, and we're going to take a pretty quick tour through the cell. In future years, if you're a biology or a biochemistry major, you'll take a whole semester course just looking at cells. So this one lecture today is really just a very brief tour through the cell. We got to begin our study of the cell by talking about when cells were first identified. This was back in the 1600s. A gentleman named Robert Hooke was able to create the first microscope where he was able to magnify the size of cells by 30 times. Very soon after that, another gentleman, Anton von Leeuwenhoek, was able to create a better microscope that could magnify 10 times more than that, so up to 300 times. By 1800s, people believed that all living things were composed of these little compartments called cells. By the mid-1800s, Robert Virchow said that all cells come from pre-existing cells. Some people believe that another gentleman, Robert Remak, came up with this idea first, but Virchow is usually the one who's given credit for it. So by the 1800s, we had really come up with the cell theory, which stated, remember, theories in science have both a pattern and a process to them. The pattern part of it was that all organisms, all living organisms, are made of cells. And the process part was that all cells come from pre-existing cells. So if we take a look at modern microscopy, there are different types of microscopes that scientists use to study cells. In the top half of this slide, we're looking at, over on the right-hand side here, this is a light microscope, and these are the kinds of microscopes that you've been using in the lab, where you're able to look at cells. You can probably magnify things up to about 1,000 times, so they're a little bit better than the ones that Hooke and Leeuwenhoek came up with. But here we see three different examples of images that we might see in a light microscope. On the left, we see some muscle cells. In the center here, we see some pancreatic cells. And on the right-hand side, we see neurons. One of the first things you're going to notice that we'll talk about in a little bit is how different all of these cells look. But we really can't see inside the cells very well. We can't see any of the structures that are inside, called the organelles. To do that, we need higher powered microscopes, and there are higher powered microscopes that can magnify to a much greater extent. The two that I'm showing examples of here are the scanning electron microscope, which is called the SEM, or the transmission electron microscope, the TEM. Over on the left-hand side, the image shows you that the scanning electron microscope really gives you a pretty good image, a surface image of a cell, gives you that 3D look. Here on the left, we're looking at the surface of a cell that has lots of these finger-like projections at the top called microvilli, and we're seeing the surface of them that makes it look like this sort of brush-like structure. On the right-hand side, we're looking at a transmission electron microscope that actually allows us to look inside the cell to see some of the subcellular particles or organelles that would be present inside the cell. Again, we can see those microvilli, but now we're looking at them from the inside. So cells are pretty amazing structures. They're able to be these independent little packets of life, and all organisms are made up of these packets. But each cell, even though it works in conjunction with the cells near it, has to be able to maintain its own proper internal environment. So the question is, how can they do that? How can they function as separate independent entities? And the reason are threefold, really. The first reason is because of their size. Cells are small, and because they're small, they have a larger surface to volume ratio than something that would be even larger. So this picture over on the right-hand side sort of gives us an idea of this. If we look at the cube on the left, the volume there is the same as the cube on the right, but the cube on the right has been broken down into much, much smaller size blocks. And these blocks each have their own separate surface area surrounding them. So each of those blocks can control what's inside its environment far better than this large block can. This has something to do with the fact that each of these individual blocks has their own surface area, or in a cell that would be its own plasma membrane that surrounds it and controls what can come in and out of that cell. We call this selective ability to regulate what comes in and out of the cell the fact that the cell membranes are semi-permeable. Now, this formatting gets really messed up on these slides when I make videos on them for some reason, but this word right here is semi-permeable, okay? Semi-permeable. The other thing that gets messed up is, looks like in these titles, it's supposed to say cells and homeostasis. Again, homeostasis is the ability for cells to maintain a proper internal environment. This should say environment. Okay, so the fact that these cells have this plasma membrane that is very selective, it's semi-permeable, allows these cells to decide what can come in and out of these cells and help maintain that internal environment. The other thing cells have that allows them to do this is that they have what's called organelles. Just like we have organs in our body, which are responsible for carrying out very different functions, cells also have organelles. So organelles are these subcellular compartments that can carry out very special functions or activities. And we're going to talk about many different organelles today. Another thing to remember when we're talking about cells is that form is function. This should say form is function. Just like anything in biology, you'll see some very specific specializations of cells that allow them to carry out special functions. For example, these are called microvilli, these little projections. And we saw this image earlier when we were talking about transmission electron microscopes. But microvilli are specializations on the surface of a cell. They're outfoldings of the cell surface that allow it to not have this flat surface, but increase what we call the surface area of that cell. And when it does that, it allows that cell to maximize its ability to absorb things from the outside world. If you have one surface that is just completely flat, and you have another surface that is all folded up like this, there's greater surface area there for that cell to have contact with the outside world and will have a better ability to absorb things from the outside world. So that we'll see a lot of different specializations like this in cells that allow them to perform specific functions. When we look at the size and the shape of different cells, we'll see there are lots of different forms for lots of different functions. So as we just mentioned before, some cells may have this surface that's in contact with some environment that it wants to absorb things from. So this is an example of an intestinal cell. There would be another cell right next to it here and another one over on the other side. But the surface of it here is free, and it's going to be facing the contents of our intestines so that when we need to absorb nutrients from what we've digested, they'll be able to be absorbed through that surface that has all of those extensions on it, giving it a good surface area for absorption. These other cells are different. Over on the left, we have a sperm cell, and it has a very specific function. Its function is to deliver the DNA that's in this head region of the sperm to an egg. And so it's got the DNA packed in there, and then it has this long whip-like tail that's going to allow it to swim so that it can get to where it needs to go. So it has a very specific form to be able to perform a very specific function. The next one in the middle here is a nerve cell. This nerve cell has a specific function of transmitting information from one area over long distances to somewhere else. So its shape reflects that. Its form determines its function. And finally, as we mentioned before, this is what we refer to as an epithelial cell. It's an intestinal cell, but it's one that allows it to be in contact with contents that's inside the intestine that would be here. And we have the cell which can increase. Now we're going to talk about two broad categories of cells. We're going to group all different types of cells into two main categories. One is called prokaryotes and one is called eukaryotes. Prokaryotes, which are shown on the left, are very small compared to eukaryotic cells. Prokaryotic cells are things like bacteria, and they have no membrane-bound nucleus. That's very critical to their definition. Prokaryote means before a nucleus, so they don't have a membrane-bound nucleus. They do have DNA, but it's contained, as is shown here, this purple stuff is just the nucleoid region. It's not a nucleus, so there's no membrane that surrounds it. Eukaryotes, on the other hand, do have a center part of the cell that's bound by membrane that's called the nucleus, and that's where the DNA is located. It's sort of thought to be the central controller of the cell because the DNA really decides all the different things that go on in the cell. Prokaryotic cells are also smaller than eukaryotic cells, and they have very little to no membrane-bound organelles inside. What's an organelle? Well, if you look in the eukaryotic cell, there's extensive organelles, membrane-bound organelles. You can see all this purple here and down here, the blue. They're like we have organs in our body. The cells have organelles. They're different compartments that carry out different jobs, and we'll learn all about those different jobs in a minute. The eukaryotic cells also have quite extensive cytoskeleton, and the cytoskeleton is a series of fibers that exist in the cell, and they give it shape, they give it structure, they give it strength, and they allow it to move. So prokaryotic cells do not have extensive cytoskeleton. So those are like the main differences between these cell types. Here's just a table that lists all those differences between prokaryotic and eukaryotic cells. Prokaryotic cells are the cell types are bacteria and archaea. So that would be prokaryotic here. I have to draw a little sign there and say prokaryotic or prokaryotes. Prokaryotes, you could say they're prokaryotic. And eukaryotes would be like the cells that we have, either in animal cells or plant cells would be good examples of that. And as you can see in this table, the location of the DNA is different in the two cell types. In prokaryotes, it's in this nucleoid region. There's no membrane surrounding it. In eukaryotes, the DNA is located in the nucleus. In terms of internal membranes and organelles, prokaryotes do not have very extensive membranes. They only exist in some types of bacteria that are photosynthetic. Eukaryotes, on the other hand, have very extensive organelle systems, including membrane-bound organelles. Again, the cytoskeleton in prokaryotes is very limited, whereas in eukaryotes it's extensive, and the overall size, usually the prokaryotes are much smaller than eukaryotic cells, and eukaryotic cells are much bigger. If we start looking at some of the eukaryotic cells, we mentioned animal cells and plant cells. They share a lot of very similar characteristics. They have a lot of the same organelles, and we'll talk about all these different organelles later. For example, they both have a nucleus. They both have things called mitochondria. They both have things called endoplasmic reticulum and Golgi apparatus. But there are some differences, and you can see some of those differences in these pictures. For example, you can see that the plant cell has a very tough outer cell wall. That's something that doesn't exist in animal cells. Both of them have a plasma membrane that surrounds the cell, but only plant cells have this tough outer cell wall. In addition, plant cells have a large central vacuole. That large central vacuole, we'll find out later, has lots of different jobs, but the animal cells don't have those. Plant cells also have these chloroplasts, the ones shown in green here. We'll talk a lot about chloroplasts later when we talk about photosynthesis, but those are, again, something that animal cells don't have. So now we're going to begin to talk about some of these organelles, some of the subcellular compartments. The nucleus is the first one we're going to talk about because it's so important. It really is the control center of the cell. It's where all the DNA is located. DNA, which, as we already know, is a nucleic acid, it's a double-stranded piece of DNA, is associated with some proteins in the nucleus. And together, the DNA plus proteins makes up something called chromatin. So the DNA is associated with proteins, and together they make up chromatin. The chromatin is going to be highly organized into chromosomes, and we'll talk about that a lot more later this semester too. Surrounding the DNA is something called the nuclear envelope, and the nuclear envelope has a double membrane to it. We'll look at that a little bit more later. But the double membrane protects the DNA inside of the nucleus, and it's got these small pores, little channels that you can see, all these little pink guys all over this nucleus, are channels that allow transport of material to and from the nucleus. So in the nucleus, as I mentioned before, the DNA is organized. The DNA is associated with proteins to make up that chromatin, and together all of the chromatin is separated into 46 separate pieces called chromosomes in humans anyway. Different organisms, different species have different number of chromosomes, but humans have 46 chromosomes in their cells. That's about 2 meters of DNA. Isn't that amazing? So
the chromatin is then organized, as I mentioned. Over on the left-hand side of this picture, you can see that the DNA, which is shown in blue here, is wrapping itself around these balls. These balls represent proteins that are called histone proteins. And there are 8 histone proteins that make up something called a nucleosome. And the DNA wraps itself around that to help organize the DNA. What we're going to find out later is that those histone proteins also help regulate how the DNA is read, and we'll talk about that more later in the semester. Over on the right-hand side, we also see DNA. In this picture, it's shown in red, and it's wrapped around those histone proteins shown in green. And then those structures are then wrapped even more tightly to make something called a 30 nanometer fiber. That's wrapped even more tightly to make a much more organized piece of DNA. So ultimately, it's highly, highly condensed into the structure which is shown below, which is called a chromosome. The next organelle we're going to talk about after the nucleus are ribosomes. Ribosomes are structures that are really important in our cell because they make proteins. We talked before that the building blocks of proteins were amino acids. So we can draw a couple of amino acids here. Here's one, and this is going to be its variable group up here. Here's another one, and it's going to have a different variable group coming off of it. Okay, here's another one. Coming off of our amino acid here. And we know all of those amino acids have to be linked together by something called peptide bonds. Remember, that's that condensation synthesis reaction to make a polymer. That polymer is a protein, and ribosomes are the machinery that get that job done. They're the ones that link those amino acids together to make proteins. Ribosomes can be found freely floating out in the cytoplasm of the cell, which cytoplasm is all that space, the watery space inside of the cell that isn't one of the organelles. They can be freely floating there, or they can be stuck on a membrane. When they're stuck on a membrane, like is shown here, these little rough dots here that we can see on the membrane, it's called the rough endoplasmic reticulum. The rough endoplasmic reticulum has these ribosomes that are along there.
So what role do the ribosomes play? The ribosomes actually play the very important role of creating proteins, and they do this by linking together amino acids. So we've talked in the past about amino acids are the building blocks of proteins, but they have to be linked together in that polymerization process. Remember that condensation synthesis process we talked about before? Well, that's the role that ribosomes play. So again, the formatting is a little weird here, but the free ribosomes, again, are going to be those that are just found in the cytoplasm. And then we also have membrane-bound ribosomes, and they play different roles. They both are going to be creating proteins, but free ribosomes create proteins that are going to stay inside the cell, in the cytoplasm, whereas membrane-bound ribosomes are going to create any of those proteins that need to be exported or secreted from the cell. They also create any proteins that are stuck in membranes in the cell. So we'll talk more about that later as well. All right, so we should move on to the endoplasmic reticulum then. The endoplasmic reticulum then is this long membrane structure that's found throughout the cytoplasm of cells and in these eukaryotic cells. There's an inside of these membrane-bound compartments called the lumen, or the interior part of them, and that lumen plays a very important role in that protein synthesis and secretion process. The endoplasmic reticulum can be in 2 varieties, though. We've already mentioned the rough ER, or the RER, that has the ribosomes on it. But there's also another part of it that doesn't have ribosomes that's referred to as the smooth ER, or the SER. So, and remember, ribosomes are what make the rough ER rough. So if we look at the endoplasmic reticulum, we can see that there are 2 different parts to it. The part on the right, shown in this transmission electron micrograph, shows those membrane-bound networks of membranes that have the ribosomes studded along there. On the left-hand side, we see the smooth ER, and that's where there are no ribosomes attached to it. The smooth ER tends to be more tubular. If you look up the diagram at the top, more tubular processes, whereas the rough ER is more like flattened sacs that we see here, more like pancakes. All right, so we talked about the role of the rough ER, but what's the role of the smooth ER? Well, the smooth ER is going to play different jobs in different cell types. They can do lots of different things. It can be responsible for making lipids as well as carbohydrates. These could be phospholipids, cholesterol, steroids, hormones, all different types of lipids or carbohydrates. It also can be a place of storage. It can store calcium ions. It can be a very important place for detoxifying chemicals. So there are enzymes that are present here that will help to detoxify some dangerous chemicals. And finally, it can be a place of glycogen storage and breakdown. So if you have glycogen that's been stored, these smooth endoplasmic reticulum have enzymes in there that can help break it down so it can free up the sugar that needs to be released back into the body. The rough endoplasmic reticulum, and this is a beautiful image of one. You can see how extensive that studding of the ribosomes along the endoplasmic reticulum can look. So these are flattened stacks of membranes, and again, their job is to create proteins that are going to be secreted from the cell or membrane-bound proteins. And they also have important enzymes in them or machinery that can add sugars to some of the proteins. When sugars are added to proteins, this process is referred to as glycosylation. And remember, when we talked about what links different sugars together, it's called the glycosidic linkage. So anytime you hear glyco, you know it has something to do with sugars. So glycosylation is the process of adding sugars to the proteins. So when the proteins are created by the ribosomes on the rough ER, those proteins may be glycosylated when they're inside that lumen or the inside part of the rough endoplasmic reticulum. All right, so this is just sort of an up-close view of a ribosome making a protein on the rough ER. The first thing that's going to happen, we haven't talked about this process at all, but ribosomes create proteins by reading another molecule in the cell that's called an RNA. We're going to talk about this much more extensively later in the semester. But when the ribosome realizes it has to make a protein, there's a sequence at the beginning of the protein as it's made, a series of a number of different amino acids that tell that ribosome, hey, this is the type of protein that's going to be secreted from the cell. So that ribosome knows that it needs to go and dock onto the rough endoplasmic reticulum. So it takes itself over to the rough endoplasmic reticulum, and those first few amino acids that start being created in that protein are referred to as the signal sequence. The signal sequence is then going to be recognized by another protein called the SRP, and that stands for the signal recognition particle. So the signal recognition particle is like this receptor that recognizes the signal sequence. Then there is a channel on the endoplasmic reticulum, this purple structure, that's called the SRP receptor. So this is a receptor that's going to recognize not only the SRP, but the signal sequence on that ribosome, and it acts as a docking station for the ribosome to come and put itself right down onto the rough endoplasmic reticulum. As I mentioned, this docking station is also a channel, so the protein that's going to be created by that ribosome can now be transported inside to the inside of the rough endoplasmic reticulum, where it finishes being made by the ribosome, and then it can fold into its proper final structure. Remember, form is function, so if the protein doesn't fold properly, it can't perform its function, and we'll talk more about that. So here's just some fun facts you should know about the rough endoplasmic reticulum. The proteins are created there. They're also modified once they're inside the rough endoplasmic reticulum, where they can have sugars, carbohydrates added, or maybe lipids added to them. There are also other proteins inside the rough endoplasmic reticulum that stay there. They're not secreted, and those proteins are called molecular chaperones. Molecular chaperones oversee the proper folding of proteins as they're being created in the rough ER. Any type of misfolded protein is going to be transported out of the rough endoplasmic reticulum and back to the cytoplasm or cytosol and degraded by structures called proteasomes. So proteasomes are places where misfolded proteins or misbehaving proteins are going to be destroyed. And finally, proteins that are going to be processed and secreted from the cell need to be transported to other compartments inside the cell before they can be exported finally from the cell. The way they do this is that they are transported in something called a transport vesicle, which also is a membrane-bound compartment. So if we look at the rough endoplasmic reticulum, we have it looking like this, and it's a series of stacks of proteins, or of membranes, and it's got the ribosomes all over it. Okay, and let's say we have proteins that are inside, it's in blue here, inside the rough endoplasmic reticulum that need to be transported. Well, what's going to happen is a little piece of the membrane that surrounds the rough ER is going to sort of bleb off, pinch off. Oops, it's supposed to be in red here. So a little piece of the membrane is going to pinch off that has the protein inside of it. That's called a transport vesicle, and that vesicle is going to then go to another compartment that we'll talk about in just a minute. That's called the Golgi apparatus. Sorry, I'm doing it in blue here. And that membrane is going to then fuse with the membrane that's surrounding the Golgi apparatus and dump the content inside to the Golgi apparatus once that membrane fuses. The Golgi has a different job, and other vesicles will bleb off of the Golgi with that content inside and transport that material to be secreted from the cell. So let's look at that a little further. This is a diagram showing that pathway. At the top here, we have the rough endoplasmic reticulum. What they're showing is the red dots represent the ribosomes. The protein that's being created inside is shown in purple here. So you see it, they're calling it a glycoprotein. It's a protein that's been created and a sugar's been added to it. This little vesicle is blebbing off or pinching off from the rough endoplasmic reticulum, and then it's going to transport over here to the Golgi apparatus, another organelle inside the cell. It then fuses with that membrane and dumps the contents inside the Golgi. Inside the Golgi, what's going to happen is the sugars are going to be modified. They're going to be sorted so that they're put into certain transport vesicles. It's like a FedEx station. They're going to be, some things are going to be added to them, but things are going to be sorted so that they go to their proper location. Once they're done in the Golgi, the material will come out and go in these transport vesicles, go to the cell surface, the plasma membrane. It will fuse with that membrane, and when it does that, the contents inside will be exported from the cell. So let's talk a little bit more about the Golgi. The Golgi is shown here. This is a diagram of what it might look like, and this is what it might look like actually inside the cell. And we talk about different surfaces of the Golgi. The cis face, okay, is the receiving side. That's where things are transported to the Golgi. So that's the part that's going to be facing the rough ER. Once inside, there's the medial stacks of the Golgi, and then finally the trans side. The trans side is the side that ships out the contents once it's been processed properly. So cis, medial, and trans. Inside the Golgi complex or Golgi apparatus or Golgi body, it's called all those different things, the material, the proteins that are delivered there are going to have some new carbohydrates or sugars added. Those sugars can be processed and the proteins can be sorted so that they go to their proper destination. Again, once they're done in the Golgi, those transport vesicles will bleb off and take the material to where it needs to go at the cell surface. All right, just like we can export things from the cell, which is a process that we refer to as exocytosis, we can also take things in from outside the cell in a process that's referred to as endocytosis. So exocytosis is transporting material out of the cell, and endocytosis is a process of importing things into the cell. The endocytosis that we're seeing here can happen in a couple of different ways. If it's just tiny little particles that are brought inside the cell in this middle frame here, that's referred to as pinocytosis or pinocytosis. It's what we refer to as cell drinking. So just little teeny bits are taken up. If very large particles are taken up inside the cell, it's referred to as phagocytosis. Phagocytosis is cell eating. It's globbing onto a big particle, and once it's inside the cell, you can see it's inside a membrane-bound compartment, and then that material can be processed, and we'll talk about that later. Another form of endocytosis is referred to as receptor-mediated endocytosis. How this works is that there can be cell surface receptors on the surface of the cell that recognize very specific particles that are outside the cell. So if the receptor is this red structure here, you can see it has a very specific shape. It's not binding to these green balls here. It's only binding to these purple structures because it has the right shape to bind them. Those structures then can be taken up inside the cell in a very specialized fashion. The cell is saying, hey, I really want to have a lot more of those purple structures. So it can increase the amount of purple structures that it's bringing inside because it's doing it with a receptor-mediated pathway. All right, another structure or organelle inside the cell is referred to as a lysosome. Lysosomes are membrane-bound compartments that are sort of like the garbage disposals or the stomachs of the cell. They're digestive enzymes inside these membrane-bound compartments, and they allow the cell to be able to digest either things inside the cell that they don't need anymore or particles that they've brought in from outside the cell. The inside of a lysosome has lots of different digestive enzymes, about 40 different enzymes, and the pH is quite acidic, and it allows the enzymes to work under those conditions. If we see this picture here, we can see here's a lysosome, the purple structure here. It's got the digestive enzymes inside of it. And we see endocytosis occurring here. So this is the plasma membrane of this cell. You can see phagocytosis is occurring. It's engulfing this large particle here. Well, what's going to happen is the lysosome will actually fuse with this endocytic compartment, this food vacuole. And when it does that, it will mix the contents of the lysosome with the contents of that vacuole. So the enzymes are now in the presence of that food particle, and then it can break it down and digest those pieces up so that the cell can then use those pieces for whatever it needs to use it for. There's also a process called autophagy. Let's say there's some old organelles in the cell that are not functioning very well anymore and the cell wants to get rid of them. It can encapsulate them inside of a vacuole, and then the lysosome can fuse with it and break down those organelles so the cell can reuse it. Cells are really good at recycling its parts. So if it doesn't need something or if something isn't working well, it can break it down into its component parts and reuse those parts. All right, another organelle we'll talk about is that large central vacuole that we see in plants. Again, we don't see this large central vacuole in animal cells, but we do see it in plant cells. The vacuole can have a lot of different roles too. It can play the role of a lysosome, so there can be digestive enzymes in it. It can also store water to maintain the proper rigidity of the plant cell. It can store, in addition to water, minerals, food. It can also be a site of waste storage too. And through the fact that it can be filled up so full, it can provide some mechanical strength to the cell, giving it some pressure inside the cell to maintain its shape. The vacuole is that large, single membrane-bound enclosed sac inside of plants and fungi, which we see here, and the membrane that surrounds it we refer to as the tonoplast. So the tonoplast is the membrane surrounding the vacuole. Another organelle we'll talk about is referred to as the peroxisome. Peroxisomes are important structures inside the cell that, similarly to lysosomes, they break things down, but what they break down are fatty acids and other organic molecules. They have that specific job. As a result of breaking down fatty acids, hydrogen peroxide can be produced. So this, again, the formatting is a little funny here. It should be H2O2 like that. And hydrogen peroxide can be very toxic to cells. So it could kill the cell off immediately if the peroxisome didn't also have the enzyme catalase. Catalase is an enzyme that can inactivate hydrogen peroxide and split it up so that it just produces water and oxygen. So catalase makes the hydrogen peroxide inactive and harmless. So big thing to remember, peroxisomes break down fatty acids and other organic molecules, but when they do that, they create hydrogen peroxide, which can then be inactivated by catalase. All right, mitochondria and chloroplasts we're only going to touch on briefly here because we will talk about them much more extensively when we study energy transfer later in the semester. Mitochondria is the site of cellular respiration, and chloroplasts in plants are the site of photosynthesis. So we will talk about them a lot more extensively, but for now you should know that chloroplasts are able to convert light energy from the sun into stored chemical energy, whereas mitochondria can convert stored chemical energy into energy that our cells can use, which is ATP. So both plants and animal cells have mitochondria, but only plant cells have chloroplasts. If we look evolutionarily at the evolution of cells, it's quite interesting when it's thought that both mitochondria and chloroplasts came into being inside eukaryotic cells. And the theory is called the serial endosymbiotic theory. Okay, it's kind of a mouthful. Serial, you can see it down here, endosymbiotic theory. Cereal doesn't mean like your Cocoa Puffs that you're eating in the morning for breakfast. Cereal in this case means like a series. It happens in a sequence. And what happened, what it's thought to have happened is that there was a eukaryotic cell, like the one shown at the top here, that was able to endocytose, okay, this is why it's the endosymbiotic theory, endocytose a prokaryotic organism that was able to convert chemical energy from one form to another. When the eukaryotic cell took up this prokaryotic cell, it was a symbiotic relationship. And symbiosis means a relationship which is beneficial to both, okay? So symbiosis in this case means the prokaryotic cell was happy because it was protected by this much larger cell. And it was also given food sources because the big cell was able to get good energy sources for it. And the eukaryotic cell was happy because this little microorganism, this prokaryote, was able to provide a very efficient use of energy that the cell could use. So it's thought that that's how mitochondria came into being inside of a eukaryotic cell. It's thought that many, many thousands, millions of years later, during evolution, some subset of these cells that now contained these areas that we refer to now as mitochondria also took up another prokaryotic organism that was able to photosynthesize, which meant those microorganisms could take energy from the sun and convert it into chemical energy. Again, when it took it up, this cell was now able to convert light energy into chemical energy, which was very beneficial to this eukaryotic cell. So cells such as this ended up becoming plant cells. Cells such as this ended up becoming animal cells. So what kind of evidence do we have for this serial endosymbiotic theory? Well, first of all, we know that, we'll start with number 2 here, that both mitochondria and chloroplasts are similar in size to bacteria. We know that they both have their own DNA and ribosomes, so they can replicate themselves, and some of the DNA is very similar to some ancestral prokaryotes. They also have double membranes, and this makes sense if you think about, here is a eukaryotic organism, here is a prokaryotic organism, and when the cell engulfs it or takes it up during endocytosis, that prokaryotic organism, and that's one step later, and let's look at the final stage. Once that endocytosis is finished, we're going to have an organelle inside that has some of the membrane from the original plasma membrane as well as the membrane from the original prokaryote. So it's got double membranes around it, and both mitochondria and chloroplasts do have those double membranes. Here we have a picture of a mitochondrion, and you can see that double membrane. You can see the outer membrane here, and then the inner membrane, which is thrown into these folds, which are called cristae. We're not going to talk too much about mitochondria now because we will be spending a lot of time talking about them later when we talk about energy transfer and aerobic respiration. But suffice it to say that mitochondria are the powerhouses of the cell. They're like the battery of the cell. They are able to take energy in the form of stored energy like glucose and are able to convert that into an energy that our cells can use, which is the energy our cells can use is called ATP. All right, so that's the ultimate energy that the cell wants, and it starts with the forms of energy that we take in when we eat. We eat food to give us energy, and that energy is then converted into ATP.
Chloroplasts are also organelles that are involved in energy transfer, but these are found in special kinds of organisms, plants or photosynthetic bacteria, that have the ability to capture energy from the sun, UV energy, and convert that energy into a stored form of energy. So the stored form of energy it's going to create is actually glucose. So it's sort of the opposite of aerobic respiration. They're both converting energy from one form to another. In aerobic respiration, you're transferring energy from glucose to ATP. In photosynthesis, you're taking energy from the sun, UV energy. That's supposed to be a U. UV energy from light energy, and you're converting that into glucose. So glucose will then be a stored form of energy that the plant cell can use later when it needs to convert that into ATP. Okay, so the next thing we're going to talk about is actually something called the cytoskeleton. And I realize that the formatting of these slides is really bad, so it's cutting off a lot of the information. So let's write it up here. Cytoskeleton. The cytoskeleton is like the skeleton of the cell. It gives the cell structure, just like our skeleton gives our body structure, but it also, like our skeleton, allows us to move. We have muscles attached to our skeleton, allowing us to move around. The cytoskeleton in the cell also allows it to move around. So the cytoskeleton is a dense network of proteins. There are these big protein fibers, and it's a dense network of these proteins. They're going to give the cell strength. They're going to give it shape and ability to move. They're going to function when the cell divides. So when one cell becomes two cells, it's going to play a role in that. And it's going to also function to help transport material throughout the cell. The cytoskeleton is actually very dynamic, which means it's constantly changing. You can make these fibers and break them down again. So we'll talk about that in a little bit more detail in just a minute. And the three types of proteins that are involved in the cytoskeleton, the biggest ones are called microtubules, and they're actually hollow tubes. The smallest one is microfilaments, and then there's some that are kind of in between those sides, and those are the intermediate filaments. So let's start with the microtubules. The microtubules are made out of a protein called tubulin, and protein of tubulin comes in two forms, an alpha form and a beta form. The alpha form and the beta form come together to form a dimer. Dimer just means two of these proteins coming together, and those dimers fit together in such a way that they form a tubule. These microtubules are very big, they're hollow, and they're long and strong, and they form a big part of the network of the cytoskeleton inside the cell. They allow chromosomes to move during the process of mitosis when the cells are going to be dividing. There are also tracks for intracellular transport of material in the cell. And there are other structures on cells called cilia and flagella that are long extensions off of the cells, and they can move back and forth. And those cilia and flagella have long microtubules in them that allow them to move, allow the cell to move, or allow the cilia or flagella to move. So again, it consists of two forms of protein tubulin, alpha tubulin and beta tubulin, combined together to form a dimer that then links up to form this hollow tube. There are proteins that are associated with microtubules, given a very appropriate name called microtubule-associated proteins, or MAPs. And there are different kinds of maps. Some maps are structural maps, which link microtubules together to give that cytoskeleton more of a rigid structure. But then there are other kind of maps called motor maps, and motor maps actually use energy in the form of ATP to cause movement along the microtubules. The two types of motor maps that we'll talk about are called kinesin and dynein. Kinesin moves things along the microtubule towards the plus end of the microtubule. And we didn't really talk about the plus and minus end of the microtubule, but microtubules actually have a plus and a minus end. If we looked at a microtubule, Let's look at a microtubule. Here's the tubule. And dimers of tubulin are being added to one end of this microtubule. That will be considered the plus end. Dimers of microtubules might start falling off of this end as the tubule breaks down, and that will be considered the minus end. So the plus end is usually the end where the microtubule is getting bigger, and the minus end is where it's breaking down. Kinesin is this motor map that's going to attach the microtubule, and it's going to move things towards the plus end. I always remember that because I think the letter K looks more like a plus. Dynein, on the other hand, is a motor map that's going to move material towards the minus end of the microtubule. Here is a picture showing this. There's a small membrane-bound vesicle that we see right here at the top of your screen. This vesicle is attached by way of one of these motor maps to a microtubule. This motor map is moving the vesicle along the microtubule in the cell towards the plus end. If it's moving it towards the plus end, it's going to be kinesin, this motor map here. And if it's moving it towards the minus end, then it would be dynein. You can see ATP is involved in this process. Anytime you have movement in the cell, it's going to require some energy. All right, so let's talk a little bit more about microtubules because they do play such an important role in our cells. The microtubules actually grow out of an area of the cell called the microtubule organizing center. The microtubule organizing center represents the minus end of the microtubules, and then the plus end will grow out from there. In animal cells, these microtubule organizing centers are referred to as centrosomes. You don't really need to know that for now, but when we talk about mitosis later in the semester, we'll talk about centrosomes. Centrosomes are really made up of two centrioles, which are found close to each other at right angles to each other. The structure of a centriole is what we call a 9x3 structure. Why do we call it a 9x3? Well, if you look at this picture here on the right, you see these long microtubules. Each one of these sticks represents a microtubule. There are bundles of 3 microtubules together that form a circle. The bundles form a circle, and there are 9 of those bundles all together around that circle. So we call it 9x3 because there's 9 bundles of 3 microtubules together. If we look at the picture on the very right is if we're looking at the end of that series of microtubules. The picture on the left shows what it would look like if you looked at the side of it. So on the right, we're just looking at the very ends of those microtubules and how they're lined up in triplicate in 9 of those triplicates all the way around. The mitotic spindle, which we haven't talked about yet, but which is going to play a role during mitosis, will form from these types of microtubule organizing centers. All right, I mentioned cilia and flagella are places in the cell where there are microtubules also. Cilia, which are shown here at the top, and again, the formatting got a little funky on us, so let's write cilia right below this, cilia. Cilia are extensions on the surface of a cell that can flap back and forth, and allow material to move over the surface of the cell. Flagella, which are shown below in the sperm that are shown in the picture below this, are actually these whip-like tails that allow those cells to move through where they need to move through. And the cilia and the flagella have microtubules in them that move against each other so that it causes that motion. Where the cilia and the flagella are attached to cells is called the basal body. Okay, the basal body would be located here on the cilia and here on the flagella, right on the cell where the cilia and flagella come off of the cell. They also, these basal bodies also have that 9x3 arrangement, just like we saw with the microtubule organizing centers. So in the picture down here on the left, that's what the basal bodies would look like. That's where the microtubules are growing out of. Once they're up inside the cilia and flagella, the organization of those microtubules is a little bit different. We call it 9+2 instead. What does that mean? That means, this is funny, this picture at the top shows the 9x3 arrangement, and at the bottom shows the 9+2 arrangement. So the 9+2 is actually 9 doublets of microtubules with 1 doublet in the center, one
pair of 2 in the middle. So 9 plus 2 is the organization of the microtubules within the cilia and the flagella. 9 times 3 is the organization of the microtubules in either the basal bodies or the microtubule organizing centers. All right, let's move on to some other cytoskeletal components. The very smallest cytoskeletal components are called microfilaments, and microfilaments are made up of actin filaments. And actin filaments are made up of, each of these balls here represents, let's draw this, each individual little yellow ball here represents a globular form of actin, the protein actin. Well, actin is going to be linked together into these long chains, and then the 2 of these chains will be linked together to make a microfilament. So you can see they're twisted together there. Where do we find actin in the cell and these microfilaments? Well, they make up something called the cell cortex, which is just below the cell surface, just below the plasma membrane of a cell. You might remember that I mentioned before, sometimes on some cells, there's just on the surface of the cell, there are these little projections that come off that are called microvilli. If we looked at a electron microscope view of this, we can see all of these fibers here. All of these fibers represent those microfilaments. They're making up that cell cortex. Not only are they here just below the microvilli, but they actually go up into the microvilli, giving them some structure also. So microfilaments play a role in giving structure to the cell. They also can play a role, this is a picture of an amoeba, and you may know that amoebas move around. They sort of send out this extension from the cell, and it allows more fluid to move there, and then the front of the cell will move forward. And the way that happens is these microfilaments polymerize. They make long chains of microfilaments, and they push out the part of the cell that's out towards this leading edge. As they get bigger, they push this part forward, allowing the cell to move. The microfilaments that are present in the back part of this cell then would depolymerize, which means they would break down, disassemble, and allow this part of the cell to move forward. So moving forward is caused by the assembly of these microfilaments, and the back part is moved forward as the microfilaments are disassembled there. Intermediate filaments are, just like their name suggests, they're intermediate in size between the microfilaments and the microtubules. They show a lot of different jobs in different cell types, so a lot of cell type specificity. Sometimes they are involved in cell shape support, sometimes they can fix organelles in place, such as the nucleus. They're more permanent forms of the cytoskeleton than the microtubules and the microfilaments are. Microtubules and microfilaments can be assembled and disassembled pretty readily, but the intermediate filaments are more stable. And again, they're going to be different types of intermediate filaments in the different cell types. This is just a picture showing you how the cytoskeleton inside the cell, so this, you can see the phospholipid bilayer here, that membrane. Okay, inside the cell there's that extensive cytoskeleton. The big purple tubes are microtubules. The black lines here represent the microfilaments that are just below the cell surface, and then the blue represents the intermediate filaments. What you can also see is outside of the cell, there's also a rich network. That's called the extracellular matrix, or ECM. The extracellular matrix is made up, makes sort of like this jelly-like consistency outside the cell. There are lots of carbohydrates out there, lots of fibrous proteins, including collagen, that are secreted from cells and allow the cell to anchor itself in this environment. Some of the fibers in the extracellular matrix are called fibronectins, and fibronectins help to organize the extracellular matrix, and they attach the cells to that extracellular matrix. There are also molecules, proteins, that are what we refer to as integral membrane proteins. They're stuck in the membrane and are called integrins. Integrins help tie the extracellular matrix to the cytoskeleton. Finally, we just wanted to point out again that cells that are plant cells have a cell wall,
whereas animal cells do not. And we can see this in this picture of two plant cells. We can see the primary membrane of the cell that's here. In here. And then we can see this very extensive cell wall that is made up of some very thick fibers. And then there are an area in between called the middle lamella that connects the two cell walls together. So cell walls are very rigid in these plant cells and give the plant cells quite their structure. Finally, I just wanted to mention some connections that occur between cells. This is looking at the inside of an animal cell. This cell might look familiar to you now. We said that this might be an intestinal epithelial cell. It's got all those microvilli that stick off the surface. But now we're getting a peek. We've ripped off part of the cell and we're looking inside the cell to see some of these connections that occur between cells. Some of these connections are called tight junctions, and tight junctions are found here towards the top of the cell. This cell might face, let's say, the inside of our intestine. So up in this area, there might be intestinal contents that are moving past the surface of these cells. What we don't want to happen is the stuff that's in the intestines to leak through in between the cells and be stuck in our body. What we do want to happen is for those nutrients and things that are passing through our intestines to go through our cells so our cells can take up what they want to take up and not take up what they don't want to take up. Remember, we said the cell surface is semi-permeable, so that membrane can decide what it wants to take up of the nutrients that are passing through. What we don't want to happen is for things to get through in between the cells. And that's what this red dotted line is representing. Something's trying to fit through there, but this tight junction is like a sealed up. It's like it's been stapled together between the two cells so that things cannot move through between them. Desmosomes are these other areas here. And desmosomes I think of sort of more like snaps. What desmosome will snap together around the membrane different cells together. So it's not completely a barrier like the tight junction is, but it's just holding. It gives the tissue some strength because it's holding one cell together with another cell. You can see there are intermediate filaments that are attached to these areas as well to give some strength inside the cell. The final kind of junction that we see between cells are called gap junctions. And gap junctions are really channels in between cells that allow a passageway for very small molecules or ions to move through. Things like calcium can move from one cell to the next through a gap junction. And that's it, guys. That's all we're going to talk about for cells. It's a lot of material. Hopefully you've learned a lot, and we'll talk more about this in class.